Hello everyone, this is Luke. Today, Gavin, Lisa, and I will be presenting a paper written in 2018 by Pasquesi et al. Titled Measurement and Finite Element Model Validation of Immature Porcine Brain Skull Displacement During Rapid Sagittal Head Rotations. Before we go into the detail of the paper, it is important to understand the anatomy discussed in this study. First of all, there are three meningeal, or membrane layers, separating the skull from the brain. The first is the dura mater, which is tightly connected to the skull. The second is the arachnoid mater in the middle, and the third is the pia mater, connected to the bumps and grooves in the brain. The top part of figure 1 labels where these layers are located. The arachnoid and pia maters are often considered as leptomeninges, or the pia arachnoid complex, abbreviated PAC. The PAC contains cerebrospinal fluids, or CSF, an arrangement of blood vessels called vasculature, and connective tissue called arachnoid trabeculi. Another important aspect to consider when studying the brain is the fulx cerebri. The fulx is a fold in the dura mater layer that separates the left and right hemispheres of the brain. This particular area has different material and physical properties than other areas of the brain, so this must be considered if modeling the full brain, as you'll see later in the study. The fulx is represented by the light blue material in figure 1. Another part of this anatomy that needs to be incorporated into a model is the parasagittal bridging vein. This vein can be the cause of extraaxial hemorrhaging when torn, as shown in the bottom left part of figure 1. Lastly, before discussing the paper, it's important to be familiar with the regions of the brain shown in figure 2. It should be noted that the anterior and posterior are the front and back of the brain, respectively, and the superior and inferior are the top and bottom, respectively. Even though this study is not on humans, the anatomy described here also applies to 3 to 5 day old piglets. The motivation of the study stems from the fact that recent studies have only started to take properties of the PAC into account when modeling the brain. As Dan and Maggie described in their video, this lab has previously examined the effects of rapid axial head rotation of piglets, as shown on the left of figure 3. These results from the study were used in another study to create a finite element model, which ultimately helped determine that linear elastic spring connectors could be used to represent the area between the skull and the brain. This conclusion allowed the study to be able to predict the occurrence of extraaxial hemorrhaging. However, it was clear that these results are specific to the axial plane of rotation, so with this in mind, the study we're discussing today examined rapid head rotation in the sagittal plane, shown on the right side of figure 3. The geometry and material properties of the PAC, bridging vein, and fulx are considered. First, the group creates a physical setup to determine mechanical behavior. Lisa will now discuss this aspect of the study in detail.
For the physical model, 3 neonatal female piglets were used to test sagittal plane brain movements during rapid head rotations. The piglets were euthanized, decapitated, and all exterior soft tissue and the mandible were removed. The sagittal plane along the midline of the skull was used for this experiment, and the meninges and brain were left intact. The right side of the brain was used for each sample. The schematic of the canister is shown on the right. The brain was placed into an aluminum pan with PMMA potting resin that was secured with screws. 24 to 48 dots were applied onto the exposed surface of the brain, and then 6 to 16 markers were applied to the skull and PMMA to assess rigid body motion. Silicone caulk was used to provide a seal around the brain and between the skull and acrylic cover plate. The lubricant was used to minimize friction between the plate and the brain, as well as to prevent air bubbles from disrupting the experiment. The brain was kept under moist conditions by periodic application of PBS, and the testing was performed within 6 hours of euthanasia. The specimen canister described in the previous slide was placed onto a sidearm that converts linear motion into a 65-degree angular rotation, called the HYGE device sidearm, and that's shown in the right in the schematic. Video recording was performed with a high-speed camera at 2,500 frames per second. 2 angular rate sensors were used to measure the angular velocity throughout the experiment. The markers on the periphery of the brain were matched to the nearest rigid surface marker to assess brain displacement relative to the rigid skull throughout the rotation. This was used because it would give the best estimate of the brain surface displacement relative to the skull. The image on the right shows a frame from the video recording that shows the brain dots around the entire periphery in part A and along the superior portion of the brain in part B. The researchers used 50 frames of video pre-motion to calculate the distances between the pairs and to develop a baseline to compare to the displacements after rotation. The displacements from these 50 frames were extracted as error measures. The maximum relative displacement achieved by each dot pair during rotation was extracted over experimental frames. Wilcox and sine ring tests were used throughout the experiment to determine significant differences between max displacement and the pre-motion errors. These results can be found in Table 2. The table at the bottom of the screen shows the averages for the peak angular velocity, acceleration, deceleration, and average maximum brain skull displacement, or BSD, that are averaged for each specimen, each rotation, and overall. There were no significant differences in the BSD measurement errors between the two rotations, shown in the rightmost column. However, there was relative translational differences between the two rotational events, the second rotation having a greater maximum displacement. This can be due to the possible damage to the tethering leptomeninges between the brain and skull from a previous rotation. There was a significant difference between these values, but more consecutive tests should be performed to determine if increasingly greater displacements occur with sequential rotations. The brain-skull dot pairs were placed around the periphery of the brain from the inferior posterior to the superior posterior. From this graph, there was increased maximum displacement near the brainstem and foramen magnum, or the inferior posterior, as well as near the superior surface, or the superior mid to posterior. The in situ model allowed for researchers to analyze brain-skull displacements during head rotations. They found that these displacements are direction-dependent and that the measurements in the sagittal direction are lower compared to the axial direction, shown in the box and whisker plot to the right. This can be due to the irregular geometry compared to the axially sectioned brain. There were a few measurement limitations that the researchers discussed, including the coarse resolution and frame-to-frame discrepancies. There was also statistically significant data that showed potential damage between rotations, but these cannot be definitively evaluated unless future studies are performed. A more in-depth analysis of the discussion and limitations will be evaluated throughout the discussion board. Overall, this information was valuable for modeling the brain-skull interface. These results helped to develop the brain-skull boundary condition for the finite element model that Gavin will go through now.
In this section, I'll be talking about the study's finite element model. The FEM parameters were iteratively tested to compare the validity of the FEM model to experimental values. FEM geometry was taken from a model by Coates et al. Of a complete piglet brain and skull. Pasquesi's group sagittally transected the model, keeping just the right side. This image is of the sagittal section, in which the brain is in blue, the falx cerebri is in yellow, and the skull is in green. Something to note is that they created the meshes based on node locations corresponding to the placement of markers in their experimental model. This let them create a FEM relevant to their exact experiment. The brain elements are represented by homogeneous, isotropic, and nonlinear hyperelastic material according to a first-order Ogden strain energy density function. This model is described by this equation, where lambdas represent the principal stress ratio, alpha is a nonlinear tissue-specific parameter dependent on strain magnitude, and mu of t is a shear relaxation modulus function. The shear relaxation modulus is described by mu sub 0, which represents the brain's shear modulus, c sub I, which represents the relaxation moduli, and tau sub I, which is the time constant. Moduli values were selected based on existing literature. The falx material was modeled as isotropic and linear elastic, while the skull was considered a rigid body. On the right is a table of material properties and their literature sources. Note that they tested a range of brain-skull connector stiffness to adjust their model. In terms of boundary conditions, the brain, falx, and skull were made distinct, not allowing translation of one material through another. Brain nodes on the sagittally transected surface were allowed to slide within the cut plane. The brain and skull were joined with linear elastic spring connectors to simulate connective tissues. Bridging vein connectors were also dotted throughout the brain-skull interface to examine their tethering effects. These are elements that can only support loading and tension. Simulations were run in Abacus using angular velocity and time inputs that corresponded to their experimental model. They primarily looked at how the maximum relative displacements of FEM nodes compared to displacements of physical markers under different brain-skull connector stiffness. A values represent the relationship between the FEM node and experimental model marker displacements at different regions of the model. Pasquesi's group settled on a stiffness of 46 newtons per meter because it resulted in A values closest to 1, indicating a near 1-to-1 relationship between experimental and FEM displacements. Interestingly, when they used this value and implemented bridge vane behavior, they continue to see similar behavior between displacements, indicating that bridge vane behavior may not play as significant a role as they had initially thought. Through finite element modeling, the group concluded several major findings and limitations. They found that the optimal brain-skull interface stiffness for sagittal transection models is 46 newtons per meter. Note that this is substantially less than axial transection FEMs that determine a stiffness of about 3,000 newtons per meter. They also noted that bridging vanes do not dominate the mechanical tethering between the brain and skull, suggesting that further examination of other connective tissue is necessary. Their study was limited by considering only peak angular velocities of 52 radians per second in accordance to previous axial rotation transection studies. The model would have to be retuned to include other peak angular velocities. The study had determined that the brain was homogeneous and isotropic, however other studies had confirmed that the brain is actually not homogeneous, consisting of differentiated gray and white matter, and shear moduli direction is not known to be isotropic. Additionally, the study also noted that computed tomography images don't always translate well to FEM models. They were primarily concerned about inaccuracies in the geometric merging between the cerebellum and the brainstem. Also, this model featured a completely smooth brain, when we know that the brain has ridges and bumps on its surface. The study was concerned about the discrepancy in the brain mass, as the experimental model did not have a mass brain, however the FEM model did. A different mass brain could have led to different deformation profiles. The study placed bridging vanes based on literature of human adult cadaveric models instead of pig models. Additionally, there is notable room for improvement in the bridging vane mechanics, such as resting state tension and pre-stress behavior. Finally, the study speculated that there could have been better models for the brain-skull boundary condition instead of using linear elastic connectors for sagittally plane transected models.
Ultimately, the objective of the study was met, as sagittal brain-skull displacement was measured with newborn porcine skulls that were subject to rapid rotation in the sagittal plane. In addition, boundary conditions were determined in order for this rotation to be modeled using finite element analysis. This study confirms what was predicted, in the sense that it shows how directional differences in rotation will result in different brain-skull displacements. This observation can soon be translated into future studies. In particular, finite element models can start to be considered for the whole head of newborn piglets. Using the whole head will allow researchers to estimate elongations in the parasagittal bridging vane, which will provide deeper insight into the cause and likelihood of extraaxial hemorrhaging. In the long term, these studies can be used as a guideline for understanding extraaxial hemorrhaging in humans. Thank you for listening, and we look forward to hearing your thoughts in the discussion board.